Summary of SARS-CoV-2 life cycle. In this video, we will summarize the life cycle of SARS-CoV-2 virus in 13 steps. Step 1. The life cycle of SARS-CoV-2 virus begins with entry of virus into the host cell. It involves binding of spike proteins to the ACE2 receptors. These receptors are mainly expressed in lungs, heart, kidneys, and intestines. Step 2. Binding of spike protein to ACE2 is important, but not sufficient for viral entry, which requires an additional proteolytic cleavage event. This is brought about by the host cell proteases, including transmembrane protease serine 2, abbreviated as TMPRSS2. As a result, the receptor binding domain of spike protein is separated from the fusion domain. This activates the fusion peptide present in the center of the fusion domain. Step 3. This allows the virus membrane to fuse with the cellular plasma membrane. Once fusion has occurred, the viral genome is introduced into the host cell. Step 4. Once in the cytoplasm, the viral RNA genome is translated with the help of ribosomes. The 5' side of SARS-CoV-2 genome consists of open reading frame 1 or ORF1, which is separated into two sub-open reading frames, ORF1A and ORF1B. Step 5. These are translated with the help of programmed ribosomal shifting into two large polyproteins, PP1A and PP1AB. Within these polyproteins are proteases or molecular scissors that the virus encodes. For example, NSP3 is papain-like protease and NSP5 is 3C-like protease. These proteases cleave giant polyproteins into many different proteins, each having a separate function for viral gene expression and replication. Step 6. RNA-dependent RNA polymerase or RDRP is an important non-structural protein produced from NSP12 or polyprotein 1AB. It is a part of replicase complex and plays a crucial role in the production of structural and accessory proteins. Step 7. The RNA-dependent RNA polymerase or RDRP acts on the original plus sense single-stranded RNA to produce two types of products. One product is a full-length negative sense single-stranded RNA. This RNA must be converted back to positive sense single-stranded RNA before it can be incorporated into the genome of newly formed virions. Step 8. And the other product uses discontinuous transcription to produce several negative sense subgenomic RNAs. Again, these must be converted back to positive sense subgenomic RNAs before they can be used to produce the structural and accessory proteins. Important structural proteins include nucleocapsid protein, spike protein, matrix protein, and envelope protein. Step 9. Replication and transcription complexes are separate rooms within the cytoplasm which are derived from rough endoplasmic reticulum. They are double membrane vesicles where viral replication and transcription can occur. These complexes protect viral genome from cytoplasmic sensors. In other words, most of the reactions shown in Step 7 and Step 8, including copies of viral genomes and subgenomic RNAs, take place inside these replication and transcription complexes. Step 10. By a process that is not fully understood, endoplasmic reticulum Golgi intermediate compartment or ERGC, which mediates trafficking between the endoplasmic reticulum and Golgi complex, is studded with the structural proteins like spike, envelope, and matrix proteins. Step 11. Next, the copies of viral genomic RNAs that are formed in RTCs are somehow transported out of those vesicles and coated with nucleocapsid protein. They are then directed towards ERGIC compartment. It has been suggested that an interaction between nucleocapsid protein coded genome and the matrix protein causes the viral genome to be enveloped within these vesicles to form new viral particles. Step 12. Next, there is final maturation and transport through the Golgi complex and distal smooth-walled vesicles. Step 13. Finally, the virions are transported through an exocytosis-like mechanism to the surface of the cell, where they are released to go on and either infect the neighboring cells or to infect a new host. Thank you.